Hello and welcome to the mind map of cellular respiration and photosynthesis for Year 11 biology. Okay, we're going to zoom on in. Okay, and we're going to start with the mitochondrion. Now, the mitochondrion was formed by an act of symbiosis, where an aerobic bacterium invaded a eukaryotic cell. It's the site of cellular respiration and ATP synthesis. It has an internal membrane called cristae that has a high surface area to volume ratio, and this allows more reactions to take place. It has a matrix on the inside where the Krebs cycle occurs. The internal folded membrane is called the cristae, and this is where the electron transport chain occurs. Mitochondrion houses its own circular DNA called mitochondrial DNA, and that contains 37 genes for aerobic respiration. ATP synthase is embedded in the cristae, and this makes the ATP. The fluid matrix on the inside contains ribosomes, and these are for the production of proteins. There are also granules in the matrix, and these assemble ribosomes. Alright, the first part of cellular respiration is known as glycolysis. So glycolysis means the splitting of glucose. It uses an enzyme cascade to break down the glucose, and there are 10 steps, which we won't go into. This process occurs in the cytoplasm, and it's anaerobic because it doesn't need any oxygen. It creates 2 pyruvate molecules, and pyruvate molecules are 3 carbon molecules. It also creates 4 ATP, but 2 are used, so the net production of ATP is 2. It also creates 2 NADH, and NADH is an electron and hydrogen ion carrier. Then we ask ourselves, is oxygen available yet? And the answer at the moment is no. So after glycolysis has made 2 pyruvates and oxygen is still not available, and so it's still anaerobic, and the different cell types respond differently, but pyruvate needs to be removed because glycolysis will stop otherwise. So a plant cell undergoes alcoholic fermentation to create ethanol and carbon dioxide, but this creates no extra ATP. An animal cell undergoes lactic acid fermentation. It makes only lactic acid, and again, no extra ATP are made. Then, if oxygen does become available, glycolysis has created 2 pyruvate, but with oxygen available, it now becomes aerobic. The pyruvate is shunted into the mitochondria using an alpha helix. The 2 pyruvates are converted into 2 acetyl-CoA in the matrix, which makes 2 carbon dioxide and 2 NADH. The 2 acetyl-CoA enter the Krebs cycle. The Krebs cycle is also known as the citric acid cycle. One cycle of the Krebs cycle, using the 2 acetyl-CoA, makes 6 NADH and another type of electron-proton carrier called FADH2, and then it also makes 2 ATP and 4 carbon dioxides. So at the end of the Krebs cycle, for one cycle, we've made 6 NADH and 2 FADH2s, which are the electron and proton carriers, as well as 2 ATP and 4 carbon dioxide. Now, all of the NADH and all of the FADH2 go to the cristae. The enzyme ATPase is embedded in the cristae membrane. The 10 NADH and the 2 FADH2 donate their electrons to the ATPase. The electron transport chain uses the ATPase to make ATP. The electron transport chain is also known as oxidative phosphorylation, which means adding an extra phosphate to the ADP in the presence of oxygen. The energy from the electrons pumps hydrogen ions through the ATPase. In this way, the ATPase makes ATP. Oxygen accepts the electrons to make water. The electron transport chain makes 3 ATP per NADH. Therefore, 3 ATP times the 10 NADH is 30 ATP. The electron transport chain also makes 2 ATP per FADH2. So 2 ATP times 2 FADH2 equals 4 ATP. We've just got a little summary of the production of ATP in the presence of oxygen, and we can see that it makes 38 ATP in total. Alright, we'll get to that other diagram in just a minute. Next, we've got the structure of the chloroplast. Again, the chloroplast was made by an act of endosymbiosis where a cyanobacterium entered a eukaryotic cell. The cyanobacterium has the enzyme chlorophyll. Therefore, the chloroplast is the site of photosynthesis. It has an internal membrane called thylakoids that are stacked to create a high surface area to volume ratio to allow more reactions to take place. It has a light-dependent reaction in the thylakoids as a first step, and it has a light-independent reaction that occurs in the stroma as the second step. Again, the chloroplast, because it is the result of an endosymbiotic event, houses its own DNA that contains 100 genes for photosynthesis. Chlorophyll is embedded in the thylakoids, and the stroma contains ribosomes for the production of proteins. The stroma also contains enzymes, particularly the Calvin cycle. Then we come to the first stage of photosynthesis. It's called the light-dependent reaction, or sometimes just called the light reaction. Chlorophyll absorbs the light and ejects electrons. The electrons leave the chlorophyll and make water split into 12 hydrogen ions and 6 oxygen molecules. This splitting releases electrons that go back to replenish the chlorophyll. We call this process phytolysis, which is the splitting of water using light. The 12 hydrogen ions and the 12 NADPH go to the Calvin cycle. NADPH is exactly the same as NADH in function. It transports hydrogen ions and electrons. The electrons from the chlorophyll move to the electron transport chain in the thylakoids. Then photophosphorylation takes place to make ATP at the ATPase. Then we get on to the light-independent reaction, and the light-independent reaction contains the Calvin cycle. The Calvin cycle occurs in the stroma, which is the fluid matrix inside the chloroplast. It requires the 18 ATP from the first stage, and it requires the 12 NADPH, also from the light reaction. The Calvin cycle also requires carbon dioxide, and this gets combined with Ruby P to make Rubisco. Rubisco is an enzyme with both carboxylase and oxygenase activity. That means carboxylase likes to use carbon dioxide and oxygenase likes to use oxygen. And we'll talk about this more when we talk about the C3, C4 and CAM plants. Anyway, 6-carbon dioxide is turned into 3-PGA, which then turns into 2-G3P. You don't need to know what 3-PGA is or what G3P is. You just need to know that they occur. The G3P is a 3-carbon sugar, and 2 times 3 carbon sugars are combined to make glucose, which is a C6 sugar, because glucose is C6H12O6. The anabolic nature of this sequence requires the 18 ATP made in the light-dependent reaction. Now we're going to talk about the different types of plants and their strategies. So photosynthesis needs carbon dioxide, but getting this requires the opening of the stomata, which allows for transpiration. Transpiration is the loss of water through the stomata. This water loss is a problem for plants as water is getting scarce. The solution is how the different plants attain and fix the carbon dioxide. Rubisco has both the carboxylase and the oxygenase activity. The carboxylase fixes carbon dioxide at low temperatures. However, the oxygenase fixes oxygen at high temperatures instead of carbon dioxide. The product of carboxylase is useful, and we call this carbon fixation. However, the product of the oxygenase activity is not useful, and we call this photorespiration. Plants need to solve the problem of water loss while still being able to photosynthesize. So there are 3 strategies. There are C3 plants, which are most plants, and they live in temperate areas, which means it's not too hot and there is plenty of water available. So they use Rubisco. Because it is cold most of the time, it makes 3-PGA, and this occurs in the spongy mesophyll cells. So C3 plants make glucose without any problem and aren't too worried about losing water. They have their stomata open during the day. C4 plants are plants adapted to hot conditions. They don't want to use Rubisco due to its oxygenase activity at high temperatures, so it uses another enzyme called PEP that only has a carboxylase activity. The PEP converts the carbon dioxide to oxaloacetate, which it then transports to the vascular bundle sheath cells. So it is a spatial solution. Once inside the vascular bundle sheath cells, it then exposes the oxaloacetate to Rubisco, which then converts it into 3-PGA and then onto glucose. The stomata of C4 plants are open in the morning and the evening during the day, not during the heat of the day at noon. Now, CAM plants are adapted to arid conditions. We know these to be generally cactuses, etc. Again, they use PEP because it only has a carboxylase activity. Again, it makes oxaloacetate. This time it transports it to the vacuoles inside the cells. Being in the vacuoles means it's not going to be exposed to oxygen. Once the oxaloacetate is inside the vacuoles, it then exposes the oxaloacetate to Rubisco to make 3-PGA, and then this moves on to making glucose. However, the CAM plants open the stomata at night to avoid the heat of the day and unnecessary transpiration. So now we come back to the central diagram. We can see the difference between C3, C4 and CAM plants. It all depends where the Calvin cycle occurs and when it uses its stomata for gas exchange. C3 plants have no problem with water, so they just do it during the day. C4 plants undergo the Calvin cycle in the vascular bundle sheath cell, and CAM plants gain their carbon dioxide at night, but then store the carbon dioxide in the vacuole, and during the day while the stomata are closed, the cells undergo the Calvin cycle to make the glucose. Thank you for listening. Don't forget to like and subscribe. Goodbye.